everyone, welcome to a new class. So, we were discussing our second chapter of NCERT biology, which was biological classification. So, we have discussed two kingdoms, which was kingdom Monera and kingdom Protista. So, which one is left? Today we are going to discuss kingdom Fungi. Okay. So, what were the topics that we know in general? What were the points? General points. Kingdom Fungi is a group that contains eukaryotic organisms, and they are multicellular organisms. Yes, and they are saprophytic or parasitic. I hope you remember all this. And their cell wall was made up of chitin and polysaccharides. So these were some general points we discussed. We have already discussed them. Now we are going to I'm going to explain this kingdom in detail. Kingdom Fungi. I hope you all are ready. Okay, let's start. Okay, so kingdom Fungi. So first of all, so this is a kingdom of heterotrophic organism. So what was heterotrophic? Heterotrophic means organisms that depends on other organisms for food, for energy. Like for example, we are depending on plants and animals for food, right? But what is in the case of plants? Do they depend on other organisms for food? No, they have the ability to produce food by themselves. So they are autotrophs and we are heterotrophs. So organisms that depends on other organisms for food is called heterotrophic organisms, and organisms that does not need to depend on other organism or they can prepare food by themselves, they are called as autotrophic organisms. So kingdom Fungi, the members of kingdom Fungi is heterotrophic organisms. They need to depend on other organisms. Okay, yes, that is clear. Now, our first point will be habit. So, first we will discuss the general characters of kingdom Fungi. Then we will go to the classification. Okay? Yes. Now, habit of kingdom Fungi. Fungi are cosmopolitan. What is cosmopolitan? Cosmopolitan means they are present everywhere in earth. Like almost everywhere they are seen. Okay? So, in every tropic regions, in temperate regions, tropic regions, everywhere we can see any member of kingdom Fungi. So, we call them cosmopolitan. Okay, this term means everywhere in the earth, in every tropical region, we can see members of kingdom Fungi. That's why we call them cosmopolitan. Okay, so they are cosmopolitan. They occur in air, water, soil, and on animals and plants. That means they are seen in air. We are like currently I am sitting here, there is air around me. Fungi is present. Fungal spores are present around me also. They are seen in water. They are seen in soil. They are seen in everywhere. Even though we can't see them, their spores or their presence is still there. Okay, so they are seen in air, water, soil, everywhere they are present. They are cosmopolitan. In every tropical region, in every part of the world, we can see a member of kingdom Fungi. So they are cosmopolitan. They are seen in air, water, soil, animals. Yes, why? How in animals? They cause diseases, so they are parasites. So when they cause diseases, they infect animals. So they are seen in animals also and also in plants. They infect plants. Fungi do infect plants. So everywhere they are seen. Members of kingdom Fungi is seen everywhere. They are cosmopolitan. Okay. Now they prefer grow in warm and humid places. Humid places. So this is an important point. Humid places. Fungi always need humidity to grow for their growth. Do you remember we keep food in refrigerator? Why do we keep food in refrigerator? To avoid growth of fungus. That's why we keep food in refrigerator. Because if if water content is present, it is easy for fungal growth. It is a favorable condition for the growth of fungi. So to avoid humidity, we keep food in refrigerator. Okay, so they prefer growth in warm and humid condition. So warm and humid condition is their favorable condition for growth. Okay, so let's discuss the examples for fungi. So first one is mushrooms. Mushrooms are edible fungi. You know edible fungi. Edible means eatable fungi. Eatable, you can eat them, you can have it. It's fine. So mushrooms are eatable fungi. Examples for eatable fungi is mushroom. That's an economical importance. So remember it in that way. Then toadstools. Toadstool is a type of fungi. Then yeast. Yeast is, I think, it's a very familiar fungi for you. For me it is, because it's used in making appam, making bread. We use yeast. And there is a point, it is used to make bread and beer. Okay. So yes, yeast, and you know yeast is a unicellular fungi also. Okay. Remember that. Then Puccinia. Puccinia is a disease causing fungi. It causes rust disease in wheat. In wheat plant, they cause rust disease. Okay. Then penicillium. Penicillium is an antibiotic. It is used as antibiotic. Okay. So these are the points. Examples: mushrooms, toadstools, yeast, penicillium and puccinia. Okay. The economical importance is important. Okay. Now let's go into their body organization. Body organization means how their body is made. Like what is, like it is cellular, whether it is made up of tissue, complete body organization, how their body is made, that is body organization. Their body consists of long, slender, thread-like structures called hyphae. Hyphae, long, slender, thread-like hyphae. Okay, let me draw it for you. Okay, let me draw it here. So this is their body, like thread-like structures. Fungal body is made up of long, slender, thread-like. This is a single hyphae. Thread-like structures called hyphae. Okay, hyphae. Remember this term, hyphae. Long, slender, thread-like structures. Very thin. It's like a thread. Simply, it's like a thread-like structure called hyphae. Okay, and this hyphae forms a cluster or a group of hyphae is called as mycelium. So here, number of hyphae together like a network. You can see it's like a network. It is called mycelium. Okay. So what we see in fungi is their mycelium, fungal mycelium. Okay. So hyphae is the unit. Structural unit is hyphae, and number of hyphae together forms a mycelium. Hyphae forms a cluster, or they forms a network forming a mycelium. Okay. So these names are important: hyphae and mycelium. Okay. Now, cenocytic hyphae. So, let's go into their structure in detail. So, consider this as a hyphae. This is a single hyphae. Okay. Wait a minute. Okay. If this is a hyphae, sometimes there are crow's walls or some crow's walls or some linking, some bridge formed in hyphae, some structure seen to separate the nucleus and cytoplasm. These crow's walls are known as septa. Okay, so the crow's walls seen in hyphae is called as septa. So what does this septa do? They can separate nucleus and cytoplasm. So septa separates nucleus and cytoplasm. If the hyphae has this crow's walls, they are called as septate hyphae or septate fungi. The fungi is called septate if the septa is present. Only sometimes the septa is seen. Sometimes the septa is not seen, like septa is absent. Then we will see a continuous, like this. If the septa is absent, the nucleus will be, the hyphae will be multinucleated. There will be many nucleus, and protoplasm, it will be continuous. A tube filled with nucleus. Mycelium looks like a tube, or hypha looks like a tube filled with nucleus. So we'll call it cenocytic. That is cenocytic hyphae. Hyphae are continuous tubes filled with multinucleated cytoplasm. So if hyphae does not have a septa or a crow's wall, it will look like a continuous nucleated tube, multinucleated tube. Then we will call it cenocytic hyphae. Okay, the terms are important. Hyphae is the unit. Thread-like structures called hyphae. Number of hyphae together forms mycelium. And if sometimes the hyphae may have septa or crow's walls between them, that this crow's wall will separate nucleus and cytoplasm. If the crow's walls are not present, so we will see the nucleus will be multinucleus, that will be continuous. Number of nucleus will be seen without any separation. We will call it cenocytic hyphae. Okay. And others have septa or crow's walls. Okay. Now, cell walls of fungi are composed of chitin and polysaccharides. So now, discussing their cell wall, it is made up of chitin and polysaccharide. We have already discussed that. Chitin and polysaccharides forms their cell wall. Okay? Is that clear? Yes. Now, once again, their thread-like structure is hyphae and its network is called mycelium. If the mycelium or if the hyphae does not have crow's walls or septa, it is called as cenocytic hyphae. And then at the end, we discussed that the cell wall of fungi is made up of chitin and polysaccharide. And in case of plants, it was cellulose. For fungi, it is chitin and polysaccharide. Okay? Chitin and polysaccharide. Now, their mode of nutrition, how they have food, how they get nutrients, that is mode of nutrition. So, in fungi, one thing, it is heterotrophic. That means it depends on other organisms for food. It cannot make food by itself. So, it is heterotrophic. First point. Then, mode of nutrition is in three types. It can be saprophytic, parasitic or symbiotic. Saprophytic, parasitic or symbiotic. So, now what is saprophytic, parasitic and symbiotic? First thing, saprophytic. Saprophytic means an organism depend on dead and decayed matter for food. Absorb soluble organic matter from dead substrate. So, soluble organic matter is absorbed from dead substance. From dead substance, their minerals and nutrients are taken or are absorbed by these organisms and they will get the nutrients. So, that is saprophytes. Depends on a dead and decayed matter. Okay. That is saprophytes. Now, what is parasite? Here, in saprophytes, they depends on dead and decayed matter. But in parasite, living organisms, the organism will depend on living organisms for food. So, the nutrients of a living organism will be absorbed. Minerals and nutrients from a living organism will be absorbed. And this is harmful for the host or the living organism. Okay. So, parasites will harm the host. So, in parasites depends on living plants and animals. Okay, the organism depends on living organisms. That means living plants and animals, we call it parasitic. Okay, yes. And what is symbionts? Symbiont is like an opposite of parasite. That means here two organisms are mutually connected in such a way that both are advanced. That means both are benefited. Okay. So, association with, so there is two types. Examples are lichens and mycorrhiza. Okay, symbionts, we will discuss symbionts in upcoming portion. There we have to discuss symbionts. So, in general, I will say symbiotic association means an association which is mutually beneficial for both members. So, there will be two partners. They will mutually connect to each other, forming an association which is called as symbiotic association, which is beneficial to both of them. An example is lichen and mycorrhiza. Lichen is a symbiotic association of algae with fungi. Algae and fungi associate together and live. We call it lichen. Mycorrhiza is the fungal association with roots of higher plants. You know, plants like cycas, pinus. So, with their roots, fungi forms an association which is called as mycorrhiza. Okay, is that clear? Lichen and mycorrhiza. So, association with algae is called lichen and association with roots of higher plants is called as mycorrhiza. Okay. Now, next is mode of reproduction. How they reproduce. Mode of reproduction is very important point. Mode of reproduction. There is three types of reproduction. It may be vegetative, it may be asexual or it may be sexual reproduction. So, vegetative reproduction means a vegetative part is used for reproduction. Any part of the vegetative plant body, vegetative part is used for reproduction. That is vegetative reproduction. Examples are fragmentation, fission and budding. Fragmentation means a fragment of the body part. Any fragment of from the body part, a small piece is taken and it will grow into a new individual. That is fragmentation. A fragment will develop into a individual. That is fragmentation. Fission means one cell divides into two cells. That is fission. Okay. Two or more cells. One cell divides to form two or more cells is called fission. Okay. Then last, budding. Budding means from a parent, a small bud is formed. The bud develops and grow into and detach from the parent and grow into a new individual. Like this. The bud get detached and it will develop into a new individual. Okay. This is budding. So, fragmentation, fission and budding is the vegetative reproduction seen in fungi. Okay. And then asexual reproduction is by spore formation. Spore is a structure that helps in reproduction. So, asexual reproduction is formed is by spore formation. There is three types of spores: conidiospores, sporangiospores and zoospores. Once again, conidiospores, sporangiospores and zoospores. Okay. Yes, conidiospores are formed in conidia, sporangiospores are formed in sporangia. Okay. Now, sexual reproduction. Sexual reproduction, there are three types of spores. Once again, oospores, ascospores and basidiospores. Okay. Okay, once again, oospores, ascospores and basidiospores. These terms are important. You will have to memorize them. Okay. Now, spores are formed in fruiting bodies. So, where is the spores formed? This is structures that help in reproduction. So, these structures are formed in a special fruiting body. It's a fruit-like structure in which spores are formed. That is fruiting body. And then what will happen with some abiotic factors like with water or with airs, with wind, anything like that, the spores will get detached or the spores will get dispersed. Okay, so spores are formed in fruiting bodies and different types of spores. Okay, first one was about their reproduction. There is 3 types of reproduction seen in fungi. Well, first one is vegetative reproduction, which involves fragmentation, fission and budding. Fragmentation, a fragment is formed which will develop into an individual. Then fission, a cell will divide into 2 or more cells. Cell division, simply. Then budding. Budding means a bud is formed from the parent body and it will develop into a new individual. That is budding. Is that clear? Yes. And the next point was asexual reproduction. Asexual reproduction means reproduction without any fertilization, anything like that. And asexual reproduction is by spore formation, which were the spores, conidiospores, sporangiospores and zoospores. And then sexual reproduction. Here we have 3 spores, oospores, ascospores and basidiospores. Okay. Last point was, where is this spores formed? They are formed in special structures which is called as fruiting bodies. Fruiting bodies. Inside these fruiting bodies, the spores are formed. Okay. Yes. Now, sexual reproduction. So, we have a lot to discuss about sexual reproduction. It is simple. Only the steps are important and there is a phase called dikaryotic phase. That's all. I'll explain it as a story. It's very simple. So, in sexual reproduction, there is 3 stages. 3 stages. The first stage is plasmogamy. Plasmogamy means, so consider 2 cells. 2 cells. 2 cells are fusing. So, the cells have protoplasm and nucleus. So, protoplasm and nucleus. Protoplasm is the structures without nucleus. Is the complete structure without nucleus is called as protoplasm. So, a cell have a protoplasm and a nucleus. Okay. So, the cell is fusing now. So, first what will happen? First their protoplasms. So, this is the protoplasm. First the protoplasms of the cell will fuse. Okay. Then their nucleus will fuse. So, these are the 2 steps. First one is protoplasm fusion, which is called as plasmogamy. Fusion of protoplasm is plasmogamy. Only a term. First their protoplasm will fuse, then their nucleus will fuse. And first protoplasm fusion is called as, fusion of protoplasm is called as plasmogamy. Okay. And now, that's the first point. Then their nucleus will fuse. And the fusion of nucleus is known as karyogamy. Okay, is that clear? The fusion of protoplasm is the first step, which is plasmogamy. Then karyogamy. Karyogamy means fusion of nucleus. Okay, and then meiosis will occur. In this, so after the fusion, when this protoplasm and nucleus fuses, we'll get a cell, right? Okay, the protoplasm and nucleus fused together, we'll get a cell. A new cell is formed by the fusion of the parent protoplasm and nucleus. We get a new cell. Okay. Now this cell will undergo division. The division type is meiosis. What is meiosis? You will learn it in the upcoming chapters. You will learn it. Okay. So meiosis is a type of cell division. Just remember, now this cell need to divide. Okay. So then meiosis will occur. So this is the zygote. Zygote is the first cell formed. So zygote is formed and then meiosis occur, and after meiosis haploid spores are formed. So what was spores? Spores are reproductive structures. So after meiosis, reproductive structures called spores are formed from that structure. Okay, once again, first 2 cells, we have 2 parent cells. First their protoplasm will fuse, then their nucleus will fuse, which is called as plasmogamy and karyogamy. Then after this fusion, we get our zygote. And then what will happen to the zygote? The zygote will undergo meiosis or cell division. Meiosis is a type of cell division. Okay, cell division occurs and then these structures will produce spores. Okay, this is how sexual reproduction occurs. Okay, yes. Then, two haploid hyphae of compatible mating types come together and fuse. Okay, so just like we said, two cells. So here two cells, like that means two hyphae. Two hyphae are this parents that will fuse together. So hyphae has protoplasm and nucleus. So that's what is going to fuse. Okay. So two haploid hyphae of compatible mating types come together and fuse. In some fusion of two haploid cells immediately results in diploid cell. That means in some fusion of two haploid cells immediately results in haploid cell formation. That means sometimes during fusion, just after this protoplasm fusion and nuclear fusion, which is plasmogamy and karyogamy, you will get a new cell, you will get a zygote. There is no intermediate stage. After the fusion will simultaneously occur and you will get a zygote. In some cases, in most of the cases. But in some cases, the fusion is a little bit delayed. That means after protoplasm fusion, after some time only, nuclear fusion or karyogamy occurs. Karyogamy occurs after, there is an interval between plasmogamy and karyogamy. Okay, because of this, a stage called dikaryotic phase is formed. That means, okay. So now these are our cells. These are our hyphae part that is going to fuse. So first protoplasm division occurred. So the protoplasm is fused. So this is our fused protoplasm. Protoplasm of both cells. Fused protoplasms. But karyogamy is still to happen. That means karyogamy haven't occurred. So plasmogamy occurred, but karyogamy is still suspended. That means karyogamy is to occur. Okay. So this stage where there is an interval stage with one cell and two nucleus. So there is two nucleus because nucleus is just going to fuse. So there is two nucleus within one cell. There is an interval stage because between plasmogamy and karyogamy there is a few second interval. So if this interval phase is seen, we call this interval phase dikaryotic phase. Remember the term, di means two, karyo means nucleus. So a phase where two nucleus is seen in a single cell. Okay, dikaryotic phase. Okay, in other fungi, an intervening dikaryotic stage, that means two nuclei per cell occurs. Okay, yes. Such condition is called a dikaryon and this phase is called dikaryotic phase of fungus. So what is dikaryotic phase? One cell with two nucleus. A stage in which there is one cell and two nucleus. It is an interval stage between plasmogamy and karyogamy. So karyogamy, it's a stage just before karyogamy. Okay, is that clear? Protoplasm division is occurred, but karyogamy or nuclear division is yet to occur. So there is an interval stage formed, which is called as dikaryotic phase. It is a stage just after plasmogamy and just before karyogamy. Is that clear? Dikaryotic phase. I hope it is clear. Okay. Later parent nuclei fuses and the cell will become diploid. So just after this dikaryotic phase, the nucleus fusion or karyogamy occurs and we get the zygote. Okay, is that clear? And then we will get our zygote. This is our zygote. Is that clear? Yes. The fungi forms fruiting bodies in which meiosis occurs, leading to formation of haploid spores. Okay, after the zygote is formed, then the meiosis will occur, and then from after the meiosis, the cells will form fruiting bodies. A structure called a fruiting body is formed in which spores are formed. Okay, so after meiosis, we will get new cells or new fungi which will develop fruiting bodies which produce spores, and the cycle continues. Okay, is that clear? Reproduction, sexual reproduction, there was three stages. First one was plasmogamy, that means protoplasm fusion, karyogamy, nucleus fusion, and then meiosis will occur, and then it will give rise to fruiting body. Inside fruiting body, spores are formed and the cycle continues. And there was a stage which is known as dikaryotic stage, in which there is one cell with two nucleus. It is a stage just after plasmogamy and just before karyogamy. That's all. That's it. Okay, is that clear? Sexual reproduction. Okay, so the general characters are completed. Now we are going to learn classification. There is classification, four classification. Kingdom fungi is divided into four groups: Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. Once again, Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. So this is the classification. On the basis of morphology of mycelium, morphology, that means external structure, how mycelium is made, how mycelium looks, their external features. In the basis of morphology means external structure. Okay, mode of spore formation, how these spores are formed, and fruiting bodies. There are different types of fruiting bodies and many types of fruiting bodies. So on the basis of mycelium, this external structure of mycelium, which is morphology of mycelium. So morphology means external structure. So on the basis of external structure of mycelium, the mode of spore formation, the way their spores are formed, and their fruiting bodies. Fruiting bodies are spore producing structures. It's a fruit like structure, it's a spore producing structure, which is fruiting body. And on the basis of this, we have classified kingdom fungi into four groups, which is Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. Okay, now we are going to discuss each of these groups in detail. So first group is Phycomycetes. Okay, let's start. Phycomycetes, first we'll discuss the habit, then their structure, then their reproduction. It's like that. Then if there is an examples or economic importance, like that. This is how we are going to discuss each of these groups. Okay. So first one, Phycomycetes. Found in aquatic habitats. Aquatic habitats, they are seen in water like sea, ocean, river, freshwater, aquatic. That is aquatic habitat, marine water, freshwater, in water where water is present. So they are seen in aquatic habitat, found in aquatic habitat. And on decaying wood, in decaying wood, that means saprophytes. They are seen on dead and decayed matter. They will absorb nutrients and minerals from dead and decayed organisms. They are seen in decaying woods, decaying twigs, leaves. Okay, so they are saprophytes. Okay. In moist, so decaying wood in moist. Okay, in moist, that means water content should be present. Okay, for fungi, water content is very important. Okay, and damp places are as obligate parasites on plants. So found in aquatic habitats, water content. Okay, then decaying wood, that means they are seen as saprophytes. Okay, and then obligate parasite, they are seen as parasites on plants also. Is that clear? Okay, and obligate parasites on plants. Simply once again, in aquatic habitats, they are seen in aquatic habitats as as saprophytes on dead and decayed organic matter, and then in plants as obligate parasites. Okay, yes, that's the first point. So that's their habit. Now their structure, how their mycelium, whether it is septate or aseptate, how their mycelium looks. Mycelium is aseptate and cenocytic. So there is no cross walls, there is no septa. So their nucleus is cenocytic. That means their hyphae looks like a tube with continuous nucleus. Many protoplasm is continuously filled inside the hyphae. There is no cross walls between the protoplasm. So the mycelium is aseptate, there is no septa, and it is cenocytic. Okay, cenocytic. Mycelium is aseptate. Aseptate means without septa or without cross walls, and it is cenocytic. Without cross walls, there will be continuous nucleus are present. So they are cenocytic. And now reproduction. First one we will discuss asexual reproduction. Asexual reproduction takes place by zoospores. Zoospores are motile spores. That means they can move from one place to other. They can move. So obviously they will be using a medium for movement. For example, water, something like that. Okay. So asexual, they need water, so it will be water. So asexual reproduction takes place by zoospores. Zoospores are motile spores. So who is motile spore? It is zoospores. Okay. Or by aplanospores. But aplanospores are non-motile spores. So So zoospores are motile spores and aplanospores are non-motile spores. Asexual reproduction is by motile zoospores or non-motile aplanospores. Terms are important, you will have to learn them. Okay, memorize them. Okay, it's easy. Zoospores and aplanospores seen in phycomycetes. Asexual reproduction, spores that helps in asexual reproduction. Zoospores are motile, aplanospores are non-motile. Okay. Now, spores are endogenously produced in sporangium. Sporangium is the spore producing structure. Spores are produced inside a particular structure, a bag-like structure or something like that. We call it sporangium. So, inside this sporangium, their spores are formed. And how their spores are formed? It is formed endogenously. What is endogenously? Inside their body, spores are formed. It's not outside. It's not exposed. So it is inside their body. So spores are formed endogenously inside the sporangium. So there is a fruiting body. For example, if this is his fruiting body, inside their fruiting body, the spores are present. That is endogenously present. Okay. Yes. Then zygospore is formed by fusion of two gametes. So here we are going to explain sexual reproduction. Sexual reproduction is by zygospores. Sexual reproduction is by zygospores. Zygospores is formed by fusion of two gametes. Okay, so gamete fusion is very important step in sexual reproduction. So which are the spores? Zygospores. Zygospores helps in sexual reproduction. And in zygospores, there are three types: isogamous, anisogamous and oogamous. Once again, isogamous, anisogamous and oogamous. Okay, what is isogamous? Iso means same. Gamous means something like spore, gamete, something like that. Okay, so there is something same or similar. Okay, similar morphology. So there is two spores. If both of them have same morphology, we'll call them isogamous, and both of them are motile. Two spores that are motile and have same morphology is called as isogamous. Okay, once again, both will have same morphology and both are motile. These are isogamous spores. Okay, both have same morphology, right? Yes, both have same morphology and they are motile. So they are isogamous spores. Is that clear? Yes. Now anisogamous. Here the spores have dissimilar morphology. They don't look similar. They are different from each other. One will be large and one will be small, like that. They have dissimilar morphology and both are non-motile. They are not even motile. They are not motile. They don't have same morphology. That is anisogamous. Isogamous means, iso means same. Gamous means spores, like that. Spores, gametes, like that. So, structures that helps in reproduction. That is gamous. Gamete, something like that. So isogamous means same morphology and they are motile. Both are motile. Anisogamous means different morphology and non-motile. Okay. And then oogamous. Oogamous means dissimilar male. Dissimilar. Male is motile but female is non-motile. So in oogamous, they are dissimilar. Okay, they are also dissimilar, but saying about motility, the male will be motile but the female is non-motile. And male will be large, male is small. In oogamous, male will be small and female will be large. Okay, and male is motile and female is non-motile in oogamous. Once again, in isogamous, both are same and both are motile. In anisogamous, both are dissimilar and non-motile. So one will be small and one will be large in anisogamous and they are non-motile. In oogamous, they are dissimilar, they are not morphologically similar, and the male is motile where the female is non-motile. So male can do movement or male can do more locomotion. They can move from one place to other, but female can't. They are still or they are static. Okay. So that's it. And now examples for phycomycetes: Mucor, Rhizopus and Albugo. Mucor, Rhizopus and Albugo. These are the examples for phycomycetes. Rhizopus is bread mold. What is bread mold? I hope you have seen fungal infections or fungal mycelium formation in bread, staling of bread, stale bread, okay, fungus infected bread. Okay, so which is that fungus? It is Rhizopus. Rhizopus is a fungus that grow on bread. Okay, if moist is present, bread will get stale, right? So which is the fungus? It is Rhizopus. Okay, and then mucor. Mucor is a parasite. Parasite that grow on mustard plant. So albugo is the parasite. Mucor, Rhizopus and albugo. Mucor, Rhizopus is the bread mold. So bread mold is Rhizopus, fungus that grows on bread. That's common fungus. I think you all will be familiar to bread mold. And then albugo, it is a parasite. Okay, so that's phycomycetes. That's simple, right? First one was their aquatic habitat, and they are saprophytic, dead and decayed matter with decaying wood, yes, and they lives in moist and damp places or as obligate parasites on plants. Okay. Then about their mycelium is aseptate or it is cenocytic, aseptate and cenocytic. Then about their reproduction, asexual reproduction is by zoospores or by aplanospores. Then spores are endogenously formed, that means spores are formed inside their fruiting body. In the sporangia is the fruiting body, okay. Then sexual reproduction is by zygospore formation, fusion of two gametes fuse to form fuses, okay, that is zygospore. And then there is three type: isogamous, where the both gametes are similar and both are motile. Then anisogamous, both gametes are dissimilar, one will be large and one will be small. And oogamous, both are dissimilar and male is motile and female is non-motile. Where in anisogamous, both will be non-motile. Okay, is that clear? Yes. And then examples were mucor, Rhizopus and albugo. So now we are going to discuss our second group, that is ascomycetes. Okay, so ascomycetes are commonly known as sac fungi. They looks like a sac. They looks like a bag or sac. So they are known as sac fungi. They are saprophytic. Saprophytic means classic. What is it? It depends on dead and decayed organism for food, for nutrition. So they are saprophytic. Decomposes, they helps in decomposition, the process of decomposition, converting large organic substances into small parts decomposition, the process of decomposition. They helps in decomposition, and they may be parasitic, disease causing, that harms an organism, that depends on living organism. Yes, that depends on living organism, parasitic. Or coprophilous. Coprophilous means organism that grow in dung. Dung or excreta. Is that clear? Dung, coprophilous. So I know it is a little bit gross for us, but for them it's their habit. Okay. So once again, they are saprophytic, decomposers, parasitic or coprophilous. Coprophilous means one who grows in dung. Okay. Yes. And about their mycelium is branched and septate. So here we can see a branched mycelium. In phycomycetes, we saw a cenocytic mycelium. There was no septa. But here we can see a mycelium which is branched. Branches are formed from a part. And it is septate. That means cross walls are also formed. Septate and branched mycelium, like this. You have the structure here. Branched and septate mycelium. Okay. And now about their asexual reproduction. So their asexual spores are called conidia. Conidia are their asexual spores. They are formed in the tip of their hyphae, which is called as, the part is called conidiophore. So in the tip of the hyphae of conidia, a conidiophore, that is conidiophore, a conidia is formed. So conidia is the spore. Asexual spore is called conidia. Once again, the asexual spore of ascomycetes is known as conidia, it is formed in the tip of hyphae, which forms a conidiophore. Okay. Conidia exogenously on conidioforms. Also here the conidia is produced exogenously. That means at the tip of hyphae a structure is formed which is called as conidiophore. Exogenously, that means externally conidias are formed. Okay. The spores are formed externally. It is not formed inside. It is not endogenous. It is exogenous. It is formed outside the body. The spores are called as conidia. It is formed in structures called as conidiophores. And how? Exogenously formed. Formed outside the structure, outside the body. Okay? Yes. Now, their spores, sexual reproduction. Sexual spores are ascospores which are produced endogenously in sac like asci. So listen to the term asci. What was the name of the group? Ascomycetes. So now you know where the name came from. Okay. So ascospores are sexual spores that is formed in a sac-like structure called asci. So asci is a sac-like structure in which the ascospores are formed endogenously. That means inside their body. Okay. Inside their body, spores are formed. Okay. So ascospores are formed endogenously in the structures called as asci. Okay. Asci or asci. Asci are arranged as fruiting bodies called as ascocarps. So number of asci together forms arranged together forming a fruiting body. Okay. Is that clear? Once again, sexual spores are known as ascospores. So sexual spores of ascomycetes is ascospores and their structure is called asci, in which spores are formed. And some asci together forms a fruiting body which is called as ascocarp. Carp means fruit. Okay. So ascocarp is the fruiting body inside which ascospores are formed. And how it is formed? It is formed endogenously. It is not formed outside the body. It is formed endogenously inside the body. Where conidia spores were formed, conidia were formed outside the body. Okay. Conidia was formed outside the body and ascospores are formed inside their body. Okay. Now, examples. Examples is Aspergillus, Claviceps, Neurospora, Morels and Truffles, Penicillium and yeast. Yeast is a unicellular member. Okay, remember that unicellular member is yeast. Okay, yeast is a unicellular member. Once again, Aspergillus, Claviceps and Neurospora. Neurospora is used extensively in biochemical and genetical work. So it have medicinal value. It is used in medicinal science. It is used in biochemical and genetical work. Who? Neurospora. Okay. And then morels and truffles. Morels and truffles are edible. They are very costly and they are edible. Then Penicillium. Penicillium is a multicellular member of ascomycetes. Once again, examples: Aspergillus, Claviceps, Neurospora, Morels and Truffles and Penicillium and yeast. So the economic important ones are important. Okay, you know yeast. I have already discussed about yeast. Like we use it in making bread and beer. Okay, I hope you remember. So that's all about ascomycetes. Once again, they are known as sac fungi. They are saprophytic, decomposers, parasitic or coprophilous. What was coprophilous? Ones who grow in dung. Okay. Then as their sexual mycelium is branched and septate. Septa or crow's walls are present. Then the asexual spores are called as conidia, which is formed exogenously in conidiophore. Okay. Then their sexual spores are known as ascospores, which is formed in asci. Asci is a sac-like structure. Sac-like structure. So hence the name sac fungi. Okay. Then asci are arranged as fruiting body. Fruiting body is called ascocarp. Okay. Then example is Aspergillus, Claviceps, Neurospora, Morels and Truffles and Penicillium and yeast. And the economical important one are important. Okay. Now our next group, which is Basidiomycetes. Basidiomycetes. So okay, Basidiomycetes are normally called as or commonly called as bracket fungi or puff balls. Okay, they looks like balls. When you see Basidiomycetes, you'll feel there is a ball kept. Okay, so they looks like balls, puff balls. Hence the name bracket fungi or puff balls. So their common name is bracket fungi or puff balls, which is Basidiomycetes. Okay. They grow in soil, on logs and tree stumps and in living plant bodies as parasites. So they grow in soil, yes, and logs and tree stumps. Simply they are saprophytic. Okay. And logs means log means part of tree that has been cut off. Okay. And tree stump means the part that's still remaining in the soil. Tree stump. Okay. On logs, tree stump and in living plant bodies as parasite. They are also seen as parasites. Okay. So they are seen in soil. They are seen as saprophytes on logs of dead trees, and then they are also seen as parasites. Depends on living organisms. Okay, is that clear? Yes. And I hope it is clear. Then their mycelium, about their mycelium, their structure. Mycelium is branched and septate, just like ascomycetes. Basidiomycetes also have branched and septate mycelium. So just like them, they also have branched and septate mycelium. Crow's walls are present. Branched and septate mycelium. Septate means crow's walls or septa is seen. So they have branched and septate mycelium. Okay. Then vegetative reproduction, about their reproduction, vegetative reproduction is by fragmentation. So vegetative reproduction is seen in basidiomycetes. So vegetative reproduction is seen by fragmentation. Fragmentation means a fragment is formed or a piece is formed that can develop into a new individual. That is fragmentation. So vegetative reproduction is by fragmentation. Now asexual reproduction, there are asexual spores are not found. So in basidiomycetes, asexual spores are not found. Okay, so asexual reproduction is generally not seen. And vegetative reproduction is by fragmentation. Now sexual reproduction, sex organs are absent. So there is no sex organ, there is no fruiting bodies, there is no special sex organs for reproduction. There is no special cells, okay, there is no gametes, there is no structures like structures that helps in reproduction. There is their vegetative body that helps in reproduction. So sex organs are absent. Now plasmogamy by fusion of two vegetative or somatic cells of different genotypes. Okay, but here vegetative cells are, here sexual reproduction is absent, right? Sorry, not sexual reproduction. Sex organs are absent, but still the reproduction occurs. Here any part of the vegetative plant body, that means any cell from any vegetative cell or any somatic cell, there is no special cells for reproduction. Any cell can be used. Any cell will fuse their protoplasm. Any two cell will fuse together and undergo plasmogamy, karyogamy, thereby forming a new zygote. Okay, so plasmogamy is occurred by fusion of any two vegetative or somatic cells. Okay, so that's how their reproduction happens. And the result structure in dikaryotic. So there is a dikaryotic phase. So any two cell, any cell of the vegetative body will fuse together forming a dikaryotic stage, forming a zygote, and during the fusion the dikaryotic phase is present. What was dikaryotic phase? A cell with two nucleus. That was the dikaryotic phase. One single cell has two nucleus. That condition was known as dikaryotic phase. Okay. So yes. So as a result, dikaryotic phase is seen. That's important. Dikaryotic phase is seen in basidiomycetes. It gives rise to basidium. So this structure will give rise to basidium. Okay. Karyogamy and meiosis takes place in the basidium. So what will happen? Okay, once again I'll explain it. What happens is, first plasmogamy will occur. So there are two vegetative cells. They fuse together and plasmogamy will occur. This is plasmogamy. And then dikaryotic plasmogamy occurred, and now our cell is in dikaryotic phase. That means plasmogamy occurred, but karyogamy is still to happen. So it is in the dikaryotic phase. From here itself, the structure called basidia is formed. So in dikaryotic stage itself, the structure basidium is formed. Inside this, after forming this basidium, the next stage, which is karyogamy and then meiosis, all this occurs after that. Okay. As a result, basidiospores are exogenously produced on the basidium. So because after the formation of basidium, karyogamy and meiosis occurs, the spores that are spores that will be produced inside the basidia in an exogenous manner. Okay, so exogenously their basidiospores will be produced. Why? Because first their plasmogamy have occurred. So their protoplasm have fused. Of the any vegetative cell, their protoplasm is already fused. But now they are in the dikaryotic phase. Now itself they will form the basidium. And after forming basidium, karyogamy occurs. That means the fusion of nucleus, and then meiosis will occur. Then basidiospores are formed. So they will be formed exogenously, outside the basidium. Is that clear? Okay. So karyogamy and meiosis takes place in the basidium. As a result, basidiospores are exogenously or produced outside the basidium. Okay. The basidia are arranged in fruiting body called as basidiocarps. So these basidiums are formed on a special fruiting body which is known as basidiocarps. Once again, these basidiums are formed on a fruiting body which is known as basidiocarps. So number of basidiocarps, number of basidiums together will form a cluster or a form like a cluster or a group forming a basidiocarp. Inside this basidium there will be their basidiospores. Okay. So basidiocarp is the fruiting body on which the basidium is formed. Inside the basidium there is basidiospores. Okay. Yes. Exogenously. Okay. The exogenously means it is not inside the body. Okay. It's outside the body. It's present outside the body. It's not inside there. It means it's not their part of their internal structure. It is placed at their surface, or not surface. It is placed exogenously outside their body, not inside their body. Okay. Yes. Okay. And examples. Now examples are agaricus. Agaricus means mushrooms. You have seen mushrooms, right? So agaricus is an example for basidiomycetes. Agaricus, which is mushroom, Ustilago, smut, and Puccinia, rust fungus. Puccinia is a fungus that causes rust disease in wheat. That is Puccinia. Okay. So agaricus, Ustilago, and Puccinia. So this is all about basidiomycetes. Once again, basidiomycetes are known as bracket fungi or puffballs. Now they grow in soil, logs and stumps, and in living plants as parasites. Their mycelium is branched and septate. Yes, that means crow's walls are seen. Septa or crow's walls are seen. The asexual spores are generally not seen. Vegetative reproduction can occur by fragmentation. There is no special organs for sexual reproduction. Sexual reproduction occurs where any two vegetative cells will fuse together. Okay. And there is a dikaryotic phase. After dikaryotic phase, the structure basidium is formed. After the formation of basidium, karyogamy and meiosis occurs. So the spores, which are basidiospores, are produced exogenously. Okay. So this basidium is formed on a fruiting body called as basidiocarp. Number of basidium together forms in a fruiting body called as basidiocarp. That's all. And the examples are agaricus, Ustilago, and Puccinia. Okay. Is that clear? Let's move on to our last group of kingdom fungi, which is deuteromycetes. This is a very small group. They are known as imperfect fungi. Why imperfect? Why imperfect? That's a question. Why imperfect? We'll discuss. Okay. So deuteromycetes, they are known as imperfect fungi, commonly known as imperfect fungi. Some members are saprophytes or parasites and decomposers of litter. So some members are saprophytes. Yes, they lives on dead and decayed matter. Parasites that lives on living organisms, and decomposers of litter. You know, decomposers help in mineral cycling. Mineral cycling, that means absorbing mineral from decayed matter, decomposition. So decomposers help in mineral cycling. Then mycelium. So these were their habit. And then mycelium, about their structure, mycelium is septate and branched. Again, this is also septate and branched mycelium. So one thing, only phycomycetes have aseptate or cenocytic mycelium. All others have septate and branched mycelium. Okay. Yes. Asexual spores known as conidia. So their asexual spores are conidia. Conidia is their asexual spore. Only the asexual or vegetative phase of this fungi is known. So that is a point. So in this case, in deuteromycetes, only asexual and vegetative reproduction is seen. So this is why they are known as imperfect fungi, because sexual reproduction is absent or is not seen in deuteromycetes. They are known as imperfect fungi. Why? Because sexual reproduction is absent here. In this group, sexual reproduction is not seen. Only asexual reproduction and vegetative reproduction is seen. Asexual reproduction is by conidia, and sexual reproduction is absent. Okay. So that's why they are known as imperfect fungi. Once again, deuteromycetes are known as imperfect fungi because sexual reproduction is absent. Okay. Yes. Once perfect sexual stage. So sexual stage is considered as a perfect stage. which is absent in deuteromycetes, hence they are known as imperfect fungi. Okay. Once perfect stage of members of deuteromycetes were discovered, they were often moved to ascomycetes and basidiomycetes. Okay, so once there were some members which were discovered with sexual stage, but later they were moved to ascomycetes and basidiomycetes. So this is a group exclusively with no members with a sexual reproduction. This shows only asexual and vegetative reproduction. Okay, is that clear? And now examples are Alternaria, Coletrotrichum and Trichoderma. Once again, Alternaria, Coletrotrichum and Trichoderma are the examples for deuteromycetes, members of deuteromycetes. Okay, once again they are known as imperfect fungi. They live as saprophytes, parasites or decomposers, and they helps in mineral cycling. And about their structure, mycelium is septate and branched. Asexual spores are conidia. Okay, sexual spores are conidia. Then sexual or vegetative phases. Both sexual and vegetative phases are only seen. And once perfect sexual stage was seen, that's a point you should just remember. Okay, just remember that point. That's not so important. Examples are Alternaria, Coletrotrichum and Trichoderma. What was that point? Once there was a member which was found to be deuteromycetes which had the sexual stage, but later they were moved to ascomycetes and phycomycetes. Okay, and then examples Alternaria, Coletrotrichum, Trichoderma. Okay, so that is deuteromycetes. So with this we have completed our third kingdom, which was kingdom fungi. So we have completed our first kingdom, kingdom Monera, then kingdom Protista, and now kingdom fungi. So this chapter is about to end. Now we'll discuss our fourth kingdom, which is kingdom Plantae. So there is two chapters in next upcoming two chapters we are going to discuss kingdom Plantae and Animalia in detail. Complete chapter is about kingdom Plantae and kingdom Animalia. Here it is just an introduction of what is kingdom Plantae, what is kingdom Animalia. Okay. So first point is eukaryotic chlorophyll containing organisms. So they are eukaryotic. Yes, they have definite nucleus and membrane bound organelles. So they are eukaryotic. They have true nucleus. Karyo means nucleus. Eu means true. So they are true nucleated organisms. Chlorophyll containing, that is an exclusive, that's very important point of characteristic feature of kingdom Plantae, that have chlorophyll pigment. Okay, chlorophyll containing eukaryotic organisms. Okay, heterotrophic insectivorous plants are seen. So chlorophyll containing, they are photosynthetic. That means they are autotrophs. But there are some heterotrophic organisms are also seen. Heterotrophic means that depends on other organisms. They can do photosynthesis. Then why are they heterotrophic, right? That's a point. That's a question. Okay. But there are some heterotrophic members that captures insects. For example, insectivorous plants that captures insect and absorb food and nutrients from this insect and have food. Why, right? They can produce food by themselves. Still, they capture other organisms. Examples are Venus flytrap and bladderwort. Venus flytrap. Venus flytrap is a special plant. It can literally capture organisms, small insect. It will capture that insect and it will squeeze them and it will take all those minerals and nutrients from that organism. Okay, that's an example. Insectivorous plant, which is Venus flytrap and bladderwort. That's also an insectivorous plant. Then parasite is Cuscuta. So Cuscuta is a parasite on other plant. A plant which shows parasitism on another plant. It depends on another plant for food. Okay, so parasite is Cuscuta. Now their life cycle. They are diploid sporophytic and haploid gametophytic generation seen. Alternation of generation is also seen. So sporophytic and gametophytic generation. Sporophytic generation means spore producing generation. And gametophytic generation means gamete producing generation. Sporophytic generation is diploid. That means a multiple of 2n number of chromosomes are present. And haploid means n number. That is haploid means n number of chromosomes are present. 2n number of chromosomes are present in diploid and n number of chromosomes are present in haploid stages. And so in detail, I know you'll have a doubt that what is diploid and haploid. You can discuss it in kingdom Plantae. Next chapter where you will be learning kingdom Plantae. What is diploid, what is haploid, everything is discussed in detail. So just remember there is two stages, a diploid sporophytic, a diploid stage which is spore producing sporophytic stage and gamete producing haploid stage. Okay. So there is two generation. These are the two generation, and this generations keeps alternating with each other. So there is an alternation of generation. Okay. Okay, so just remember these points. Okay, because you have this chapter to discuss in detail, just remember these points. Eukaryotic, chlorophyll containing, there are heterotrophic organisms, and then examples and example for parasitic organism and their life cycle, which shows alternation between two generations. Okay. Okay, so next kingdom Animalia. Kingdom Animalia consists of multicellular heterotrophic eukaryotic organisms. We are the members of kingdom Animalia. We are multicellular. We are made up of number of cells. We are eukaryotic. We have true nucleus, and we have definite nucleus with membrane bound organelles. So we are eukaryotic, and we are heterotrophic. We depends on other organism for food. So we are heterotrophic. We can't produce food by ourselves. So we are heterotrophic. So once again, we are multicellular, heterotrophic, eukaryotic organisms. Eu means true, karyo means nucleus. Okay. Then holozoic nutrition. Our type of nutrition is holozoic nutrition. So what is holozoic nutrition? Holozoic nutrition, ingestion of food. We can ingest food. We can ingest means, or we can swallow, or we can eat food. That is holozoic nutrition. Okay. If you can ingest food, we call it holozoic nutrition. Okay, so that's about kingdom Animalia. So all our 5 kingdoms are completed. Kingdom Plantae and kingdom Animalia, there is separate chapters for each of those kingdoms. So 3 kingdoms, kingdom Monera, kingdom Protista, and kingdom Fungi, in detail we have discussed. Now we have a small portion, a very small portion to discuss, which is virus, viroids, prions, and lichens. This is the next small, it's a small topic. Okay, very small topic. So first, virus. I hope you all are very similar with the term virus, because nowadays we are keeping hearing, like we are hearing the term virus like many times. Like 2020 was a year which was known for a special virus. Who was it? Coronavirus, right? COVID-19. So virus means venomous or venom or poisonous fluid. So virus, virus is disease causing. So now let's see what virus is. There is a question for you. Find out whether virus is living or non-living. We'll discuss it, okay? But still, if you want, you can find it out. Whether viruses are living or non-living. Okay. So virus means, the term virus means venom or poisonous fluid. We know they are poisonous. They are disease causing, right? So the term means venom or poison. Poisonous fluid. Venom or poisonous fluid. Term virus. Dmitry Ivanovsky in 1892 discovered virus. So it was Dmitry Ivanovsky who discovered virus. Okay, 1892. In 1892, virus was discovered by Dmitry Ivanovsky. So the term means venom or poisonous fluid. Venom or poisonous fluid. Viruses are non-cellular. So they are non-cellular. They are not a cell. It is not a cell with organelles. There is nothing like that. They are non-cellular. Obligate parasite. Okay, so viruses are non-cellular, obligate parasites. So what is obligate parasite? Obligate parasite. The term obligate means an organism which cannot complete its life cycle without a host. It needs a host. Without a host, it cannot complete its life cycle. So its life is incomplete without a host. Others are free. We are free organisms. We don't need a host to live our life. Like we are heterotrophs, but we are not parasites, right? So obligate means an organism which cannot complete its life cycle without a host. A host is must. So virus is an obligate parasite. It is non-cellular. Its body is not made up of any proper cell structure, and it is obligate parasite. Okay, they can infect a cell in such a way that they will take over the machinery of the host cell. Okay, so what do they do? Like here the virus will take over the complete machinery of a cell. That means here the virus will infect the genetic material. It will capture the genetic material of the host. So in a cell, everything is determined by the nucleus. Nucleus is the brain of a cell. Nucleus determines everything. So what will the virus do? It will incorporate its genetic material into the genetic material of the host. So now the complete cell is under the control of this virus. That's how virus infect. So it will take over the complete machinery of the host cell. They are inert outside their specific host. That is, they are non-living outside the specific host. So outside the specific host, viruses are very silent. They are not doing anything. They are so good. They are just like an object. They are non-living outside a host. But just after it gets into a host, it will become a parasite. It will start its action. It will take over the control of the host cell, and it will take over the genetic material and take over the control of the complete control of the host cell. So it will become parasitic. Okay. So outside cell, it is inert, or outside a living host, outside a host, the virus will be a inert organism. Okay, inert means it will not show any response, but just inside that cell, it will show its action. It will take control of the host cell. Okay. Once again, virus, the term means venom or poisonous fluid. It was discovered by Dmitry Ivanovsky in 1892. It is non-cellular obligate parasite. That means it needs, a host is must for its growth. And they infect a cell, they take over, they infect a cell and they will take over the machinery of the host cell. Okay. Now they are inert outside the specific host. Now structure of virus, we said it is non-cellular, so there is no proper cell structure for a virus. So how is its structure? Virus is simply a protein coat with a genetic material. So a simple protein coat with a genetic material. So this is a virus. It will have a protein coat, inside it a genetic material. So it is a mere structure. A structure with protein coat and genetic material inside it, we call it virus. Okay, it's not like an organism, or there is no proper cellular structure. It is just a protein coat, and inside it will be there will be some genetic material present. The genetic material could be either DNA or RNA. The genetic material can be DNA or RNA. It can be DNA or RNA. The protein coat called capsid, made of subunits called as capsomes. So yes, the protein coat is called a capsid. Capsid is made up of units called capsomes. So many capsomes will together form a capsid. So capsid is formed by units called, the basic units of their protein coat is called capsomes. So their protein coat is capsid, which is made up of number of capsomes. Okay. Capsid made up of capsomes. And once again, the genetic material can be DNA or RNA. So there are DNA virus and there are RNA virus depending on genetic material. And one more thing, the protein coat is non-infectious. In virus, the protein coat is non-infectious. It is their genetic material which is infectious, which is harmful. So genetic material is a bring. Genetic material is harmful. Protein coat does not cause any disease, or it is not harmful. Okay. Now, these capsomes are arranged in helical or polyhedral shape. So these capsomes will be arranged in helical or polyhedral shape, forming the capsid. So, capsid is its protein coat, which is formed by basic units called as capsomes, and the capsomes will be arranged helically in helical structure or in polyhedral shape. These are two shapes, helical or polyhedral shape. The capsomes will be arranged to form a capsid. Okay. Now, examples for virus. Bacteriophage. Why we said bacteriophage? You know, bacteriophage is a virus that infect bacteria. Think about it, two microorganisms like virus and bacteria. Here, a virus that infects bacteria is called bacteriophage. Even bacteria are fed up with virus because there is a virus called bacteriophage that can infect bacteria. Okay, viruses that infect bacteria are called as bacteriophage. They are usually double-stranded DNA. So there is two types of DNA, DNA virus, double-stranded DNA virus. So once again, bacteriophage is virus that infect bacteria, which is a double-stranded DNA virus. Double-stranded DNA virus. Okay, that is the structures. Remember, capsid, capsomes, and their genetic material, which can either be DNA or RNA, and their capsid will be in a polyhedral shape. Sorry, capsomes will be arranged in helical or polyhedral shape. That's all. And an example was bacteriophage, which virus that infects bacteria. Okay. Now, in general, viruses that infect plants have single-stranded RNA, and viruses that infect animals have single-stranded or double-stranded RNA or double-stranded DNA. So that's a general point. That means plants are infected by viruses with single-stranded RNA. Okay, and animals are commonly infected by virus with double or single-stranded RNA or double-stranded DNA. Okay, that's a general point. Remember that point. Plants are mostly infected by RNA virus and animals are mostly infected by DNA viruses or RNA virus. Animals can be infected by both DNA and RNA virus. There is no difference. Okay. Viral diseases. There is some examples for viral diseases that are important. Mumps, smallpox, herpes, influenza, AIDS. Okay, these are some viral diseases. Okay, if you have some points of your own, you can write it down. Okay, viral diseases, mumps, smallpox, herpes, influenza, AIDS. These are viral diseases caused in animals. There are plant viral diseases also. There are some diseases like tobacco mosaic disease. It is a viral disease. The infected plant is tobacco plant. Okay, it is a viral disease, and there are some symptoms for viral disease. In case of plants, we are discussing some symptoms, that is mosaic formation. Mosaic means a pattern, a yellow pattern, a mosaic formation. Leaf rolling, rolling of leaves, curling of leaves, like that. Yellowing, the chlorophyll content of leaf will decrease, hence it will become yellow color. Leaves are becoming yellow color, that is yellowing. Vein clearing, the chlorophyll content decreases, the vein will become more clearer. And then dwarfing means that height will decrease. Stunted growth is suspended, growth is stopped, stunted growth. So mosaic formation, leaf rolling, curling, yellowing, vein clearing, dwarfing and stunted growth are symptoms in plants. So these are symptoms of plants which is infected by virus. Okay. So once again, viral diseases, examples for viral diseases: mumps, smallpox, herpes, influenza, AIDS in animals and some symptoms in plants. Okay. So virus is also completed. Next is viroids. So I hope you are you familiar with this term, viroid? Oh, this term has some similarity with virus, right? Viroid is discovered by T.O. Diener. The one who discovered viroid is T.O. Diener. It is smaller than virus. So viruses are smaller than virus. It is smaller than virus. Structurally, it is smaller than virus. The disease caused by viroid is potato spindle tuber. Potato spindle tuber is a disease caused by viroid. And it is smaller than virus once again. It is a free low molecular weight RNA without a protein code. So here it is simpler than virus. It is just an RNA. There is not even a protein code. It is just an RNA, an infectious RNA, which is viroid. So viroid is simply a infectious RNA. It does not even have a protein code. Okay, so viroids are just a infectious RNA without a protein code. It was discovered by who? It was discovered by T.O. Diener. And then they are smaller than virus and they have low molecular weight. That's why they are smaller than virus, right? And the disease caused is potato spindle tuber disease. Potato spindle tuber disease. Okay. Now, the next the next member is prions. Abnormally folded protein. So here we have a protein. Viroids were a genetic material. Virus had protein and genetic material. Viroid had just a genetic material. Now we have prions, which is just a protein. It is an abnormally folded protein. A folded protein. Okay, abnormally folded protein. It is prions. Okay, cause infectious neurological diseases. Oh my god, even though it is just a protein, it can cause neurological diseases. Infectious neurological diseases can be caused, which is bovine spongiform encephalopathy. This is mad cow disease in cattle. Mad cow disease in cattle, which is known as bovine spongiform encephalopathy. I know the term is a little bit big term, but still, mad cow disease, that's easy, right? Mad cow disease, commonly called as mad cow disease in cattle. Once again, bovine spongiform encephalopathy, which is known as BSE, which is known as mad cow disease in cattle. So it is a neurological disease caused by prions. Prions are protein in abnormally folded protein which is infectious. Okay. And They also cause Jacob's disease, CJD, in humans. In humans, they can cause analogy for this BSc, Jacob's disease, Jacob disease. So, CJD in humans. So, diseases are very important. It's very easy, I think. Virus is discovered by Theo Diener. It is smaller than viroids. It is discovered by Theo Diener. It is smaller than virus. Disease, potato spindle tuber disease, and then prions. Okay, and viroids are simply, it is an RNA without protein coat. And prions, it is an abnormally folded protein. It can cause neurological diseases. For example, bovine spongiform encephalopathy, which is mad cow disease in cattle. And then there is Jacob's disease, CJD, in humans. Okay. Now we have our last member, which is lichens. Okay, we have discussed lichens. Shortly we have discussed lichens. Now we will discuss lichens in more detail. Okay. It's also simple. It's very simple. Lichen is a symbiotic association. Symbiotic association, what is symbiotic association? An association which is mutually beneficial. You know, in an association there is two members. If the association is beneficial for both members, we'll call it symbiotic association. So, symbiotic association between algae and fungi, mutually useful association. It is symbiotic association. Lichen is a symbiotic association between algae and fungi. Algae and fungi. Symbiotic association between algae and fungi. So, it is a mutually beneficial association. Here the algal partner is known as phycobiont. Who is algal partner? Phycobiont. I already said that phyco is a term that is used to indicate plants, algae, organisms like that. So, algal partner is known as phycobiont. Fungal partner is known as mycobiont. Okay. So, algal partner is known as phycobiont and fungal partner is known as mycobiont. You know, the study of algae is known as phycology, and study of fungi is known as mycology. Okay. It's an extra point. If you want, you can point it down. So, symbiotic association, which is a mutually useful association between algae and fungi. Algal partner is phycobiont and fungal partner is mycobiont. Okay. Okay, and there is a point that algal partner is phycobiont and algae can produce their own food. It's a member of kingdom Plantae. Okay, so algae can produce their own food. They are autotrophic, but fungi, it is heterotrophic. Okay, we have discussed kingdom fungi. Okay, kingdom fungi members are heterotrophic. So, they are heterotrophic, they can't produce their own food. So, what is their function? Algal partner will prepare food. Algal partner can do photosynthesis, so they can prepare food for the fungal partner. And fungal partner will provide shelter and absorb raw materials for the preparation of food. Okay. So, algal partner will prepare the food and fungal partner will produce, it will give shelter to the algal partner. It can easily establish, fungal partner can easily establish themselves. So, they will give shelter to the algal partner and it will absorb raw materials for the preparation of food. And the algal partner, using all this raw material, they will prepare the food. Okay. That is their function of each members. And they are known as pollution indicators. They are very good pollution indicators. That means they will not grow in polluted area. So, if this lichens are seen in an area, that means that area is pollution free. So, they are pollution indicators. That's an important point. That's an economical importance. Pollution indicators do not grow in polluted areas. Once again, what is lichen? Lichen is a symbiotic association between algae and fungi. We have two partners. Algal partner is called phycobiont, which is the autotrophic partner, and fungal partner is known as mycobiont, which is the heterotrophic partner. Now, the algal partner will prepare the food for the fungal partner, and the fungal partner will provide shelter and absorb raw material and give it to algal partner for the production of food. Okay, and now they are known as they are pollution indicators. Why? Because they will not grow in polluted areas. So, with this, we have completed our second chapter, biological classification. So, in this chapter, we have discussed all three kingdoms in detail, and simply we have introduced kingdom Plantae and Animalia for you. And we have discussed the history of classification. Never forget that. Okay, history of classification. Aristotle, Carl Linnaeus and R.H. Whittaker. okay, and their classification, the general points, don't forget that. And then we have discussed all this with extra plus we discussed virus, viroids, prions and lichens. Okay, so I hope you all have understood this chapter completely. See you soon. Bye.